Good morning all of you. Today we will study about the structure of DNA. Let us start. As far as DNA is concerned, actually the double helical structure of DNA was first discovered or proposed by Watson and Crick. Okay, the double helical structure of DNA was first given by Watson and Crick by using the X-ray crystallography of Maurice Wilkins and Rosalind Franklin and chemical analysis data of Sargaf. Okay. So what was their hypothesis? First one is that the DNA is made up of two polynucleotide strands. You have already been taught what polynucleotide is. That means made up of many nucleotides. So DNA is made up of two polynucleotide strands which wound around an imaginary axis in a helical manner. So DNA is made up of two polynucleotide chain which wound around an imaginary axis in helical manner, forming right-handed double helix. Okay, forming right-handed double helix. The two strands are anti-parallel to each other. That is, if one strand runs in 5' to 3' direction, the other strand runs in 3' to 5' direction. Got it? So this is what we call as anti-parallel to each other. Now as you can see the picture here, there are presence of two DNA strands. There are presence of two DNA strands. If it is 5', then it will be 3', then this one will be 3' and this one will be 5'. So so this is what your anti-parallel nature is concerned. Got it? In a polynucleotide chain, there is presence of phosphodiester bond. Okay, what are phosphodiester bonds? That I will let you know in a separate video, specifically meant for phosphodiester bonds. So phosphodiester, di means two. There are presence of two ester bonds along with the association of phosphate group. So we call it as phosphodiester bonds. So there are presence of phosphodiester bonds between 5' carbon of one pentose sugar of one nucleotide and 3' carbon of another pentose sugar of another nucleotide. So the in a polynucleotide chain, phosphodiester bonds are formed between 5' carbon of one pentose sugar of one nucleotide and 3' carbon of another pentose sugar of another nucleotide. Okay, one end of the polynucleotide chain has free phosphate group. Okay, one end of the polynucleotide chain has free phosphate group attached to the 5th carbon atom. Got it? Because when you consider about nucleotide, there are presence of both nitrogenous base, pentose sugar and a phosphate group, and that phosphate group is attached to the 5th carbon atom of the pentose sugar. So one end of the polynucleotide chain has free phosphate group attached to the 5th carbon of the pentose sugar, and the other end has free OH group of 3rd carbon of sugar. That I will show you in diagram in the next couple of slides. Carbon of sugar. Okay, the nucleotides are attached to this 3' end. Remember it, the nucleotides, the elongation of nucleotides, that the elongation of DNA chain occurs by the attachment of new nucleotide to this 3' end of the your pentose sugar. Got it? So polynucleotide chain grows in a 5' to 3' direction. That is the reason why polynucleotide chain grows in a 5' to 3' direction. Got it? Now let us come to the next slide. Sugar and phosphate, they make the backbone of polynucleotide chain. So backbone of polynucleotide chain is formed by sugar, that is pentose sugar and phosphate group. Got it? The sugar is what? Deoxyribose sugar in case of DNA, and in case of RNA it was ribose sugar. Here deoxyribose sugar is found. Now the two polynucleotide chains are complementary to each other, with always purines attached to pyrimidines and pyrimidines attached to purines. So if there are purines here, then it is a rule. If there are purines here, it is a rule that there must be pyrimidines. if there is pyrimidines here, there is a rule that it must be purines. Got it? In the two strands. Now the two strands are held together by means of hydrogen bond between bases. You know, bases are present towards the inner core. They form the inner core of the DNA, and these bases are attached to each other by means of hydrogen bonds. That is, purine is attached to pyrimidine by hydrogen bond, or pyrimidine to purine by hydrogen bond. Got it? There are presence of two bonds between adenine and thymine, and presence of three bonds between guanine and cytosine. Got it? Now, sugar and phosphate backbone of both strands are located outside of helix. As I have already told you, out of the three, that is sugar, nitrogenous base and phosphate group, the sugar and phosphate, they form the backbone and they are present towards the outer side, whereas the nitrogenous bases, they form the core of the helix. That is, they are present towards the inner side of the helix. Got it? Now, as far as the dimension is concerned, the diameter of DNA is around 20 angstrom, that is around 2 nanometer, and the DNA makes a complete turn at a length of 34 angstrom. Okay, it makes a complete turn at a length of 34 angstrom, and there are 10 bases per turn. So distance between two consecutive bases is 0.34 nanometer. Okay, the angle between two base pair is 36 degree. The angle between two base pair is 36 degree. Now let us see how these are present. Now, as far as I have already told you that two strands are complementary to each other. As you can see here, the red is adenine and yellow is thymine. So adenine is a excuse me. So adenine is a purine, whereas thymine is a pyrimidine. So as far as the rule is concerned, purine should be attached to pyrimidine or pyrimidine should be attached to purine. Here you can see red, it is adenine, and here it is yellow, it is thymine. So it clearly shows that, this picture clearly shows that these are complementary to each other. Okay. Similarly, guanine and cytosine. Guanine is a purine and cytosine is your pyrimidine. Guanine is a purine and cytosine is a pyrimidine. So look at it. Guanine is represented in blue and cytosine is represented in green. Here it is guanine and here it is cytosine. So it completely shows that both the strands are complementary to each other. Now, I said that the diameter of the DNA is 2 nanometer. So how it is calculated? This one. It is 2 nanometer or 20 angstrom. Okay. Look at it. Now, if you go to the next picture, now look at it. This one is the nitrogenous base. Just let me draw a dividing line here. This is one strand, this one is another strand. This one strand, this one is another strand. In this strand, there is presence of a pentose phosphate, there is presence of phosphoric acid group, then there is presence of a nitrogenous base. Got it? Similarly, here also there is presence of a nitrogenous base, there is presence of pentose sugar, and there is presence of your phosphoric acid. Okay, phosphoric acid. Now, look how they are connected. The phosphate group and the pentose sugar, they are present towards the outer side, whereas the nitrogenous bases of both the strands, this strand as well as this strand, are they are projected towards the inner side and they form the inner core of the helix. Okay, they form the inner core of the helix. Now, as far as the two strands are concerned, this particular strand is running in a 5' to 3' direction, and this strand is running in a 5' to 3' direction. So, if it is, you know that the DNA strand grows in a 5' to 3' direction, thus this particular strand grows in a 5' to 3' direction in this way. Got it? Grows in this direction towards 3' end of the OH group. Here 3', here there is presence of 3' carbon, 3' carbon to which new nucleotides are attached. So it grows in this direction. Got it? Here also it grows in a 3' direction, 5' to 3' direction. Though these two are running in a parallel manner. Got it? So now, what are the function of DNA? In this picture also you can see the two strands are complementary to each other. Okay. Now let us see the as far as the your function of DNA. You know that DNA maintains, maintains stability due to hydrogen bonds between nitrogen bases. This DNA is highly stable because of the presence of hydrogen bonds between nitrogenous bases. Got it? And DNA is both catalytic, autocatalytic as well as heterocatalytic in nature. So what is that autocatalytic nature? That means it is able to produce itself. It is able to replicate itself. Another copy can be formed from it. At the same time, it can synthesize RNA from it. So it is autocatalytic in function as well as heterocatalytic in function. It undergoes both replication as well as transcription. Got it? And DNA has, you know that DNA has phosphoric acid in it, which makes it acidic in nature. So this particular section you can study it as what are the nature or chemical properties of DNA. This particular section we call it chemical properties of DNA. Chemical properties of DNA. This particular section is chemical properties of DNA. What are those? DNA maintains stability due to presence of hydrogen bonding between the nitrogenous bases. DNA can undergo both replication as well as transcription, and DNA has phosphoric acid which makes it acidic. So now what are the functions of DNA? Now DNA, you know, it carries all the genetic information. It is the most important vital function. Actually, it not only stores the genetic information but expresses itself and function as a director of cellular activity. That means it not only stores the genetic information, it also directs the various functions of cellular activities. So these are the two important functions of DNA. Okay, you know, have you heard that there are many forms of DNA? Those include many forms of DNA. I am not saying types of DNA, it is forms of DNA. What are those? A DNA, then you can say B DNA, okay, then Z DNA, then your C DNA. As far as A DNA is concerned, here it is right-handed, B DNA is also right-handed, C DNA is also right-handed, but remember that Z DNA is left-handed. Okay, Z DNA is left-handed. Okay, now another important thing is that remember all these things. As far as A DNA is concerned, there are 11 bases per turn. Okay, in case of B DNA, there are 10 bases per turn. What we have learned. Okay, 10 bases per turn. In case of Z DNA, there are 12 bases per turn, and in case of C DNA, there are 9 bases per turn. Got it? In case of C DNA, there are 9 bases per turn. Got it? Now, normally in a cell, B DNA is found. In case of a particular cell, normally generally B DNA is found. Now, what type of DNA is formed? It actually depends on the salt concentration in nucleoplasm. You know, nucleolus, nuclear membrane, nucleoplasm, nucleoplasm, and chromatin threads, the structure of nucleus. Inside the nucleoplasm, there is presence of chromatin threads or DNA. Now, inside it, what kind of DNA will be found? It depends on the salt concentration. In higher concentration of sodium, potassium, and cesium, in the surrounding medium, DNA exists as A DNA. When there is higher concentration of sodium, potassium or cesium, then we call DNA is found in form of A DNA. But when the surrounding medium is rich in lithium ions, DNA exists as C DNA. Got it? And in case of Z DNA, sugar phosphate backbone assumes zigzag path. Remember it. In case of Z DNA, sugar phosphate backbone assumes zigzag path. Remember that B DNA gets converted to C and A DNA during low humidity. So with this, I conclude this particular video. I hope all of you will be able to understand the structure of DNA. In the next one, I will discuss in gist regarding the replication of DNA and the process of transcription. Thank you.